Okay, I'm going to go ahead and get us started as more people are trickling in, just to make sure we have enough time for questions at the end. So it is my great pleasure to introduce our speaker today, Shaojun Wang. He is from Fudan University, and he is a PhD student there supervised by Professor Zhu. And him, as well as some of his colleagues, were the first place winners in the CAFA 5 protein function prediction that took place on Kaggle this past year. So he will be presenting their work today on GoCurator. So Shaojun, whenever you're ready, I'm excited to have you today.
Hello, everyone. Oh, wait a minute. Okay. Hello, everyone. Let's start. I am Shaojun Wang from the GeoCurator team. I would like to express my sincere gratitude to ML for protein for inviting me to share my insights on the competition. Next, I will report on today's Next, I will report on today's presentation from the following aspects. We are PhD students from Institute of Pre-Inspired Intelligence, Science and Technology at Fudan University, supervised by Professor Sanfeng Zhu. Our research interests mainly focus on bioinformatics and biomedical text mining. Prior to CAFA 5, our lab also participated in CAFA 3 and CAFA 4, which were also very helpful for improving the predictive performance of our competition. Our model for this competition is also based on our previous work. So first, I want to make some introduction about the protein function prediction. So first, what is protein function? Here we use gene ontology to describe protein function. Now, gene contains more than 50,000 biological concepts in 3 domains: molecular function, biological process, and cellular component. And protein function annotation is going to assign proper geoterms for proteins. It's obvious that proteins plays important role for our life, and a protein always has multiple functions. Therefore, a protein can be associated with more than one geoterm in gene ontology. However, knowing the function of blood proteins is difficult as the cost of wide experience is high and is also time consuming. And only less than 0.1% of protein has valid experimental functions. Most proteins still need to be explored for their functions. So designing a high-performing method to make automatic function prediction for proteins is important, which can help researchers to improve their efficiency. But function prediction is a challenging project. First, geo is a structured ontology. How to deal with the relationship in it is difficult, and every protein can be associated with many geoterms. As we all know, a large-scale multi-label classification problem is not easy. Moreover, there exists a huge variation in in the orientation. Some geoterms may have annotated many proteins, but some geoterms may only related to a little number of proteins. So in order to deal with the problems and make prediction more efficient, it's a good idea to use different sources about proteins to achieve high-performing prediction, such as amino acid sequence, protein-protein interactions, and the protein three-dimensional structure, and the protein annotation text. and so on. Later, machine learning methods can help make prediction like logistic regression, convolutional neural network, recurrent neural network, and transformer. And next, I will introduce some of my work, of our group's work, and I believe it will help you better understand our work. GeoLabel is our first work on protein function prediction. GeoLabel obtains effective features from protein sequences in different ways and trains various small models based on annotated data. Using these simple component methods, such as the baseline methods in naive and blast in CAFA, as well as sequence featured K-mer and itpro and profet methods. GeoLabel obtains initial predictions. These predictions are then integrated using a ranking model to complete the final prediction. Specifically, in the integration process, we treat the proteins as the query and each field terms as the document to be retrieved. We consider the prediction results from the component methods as protein function as proteins features, and then completing the training of the learning to rank model. We applied these methods in CAFA 3 and achieved outstanding performance, obtaining the highest Fmax in MF, BP, and CC in CAFA 3. Based on the fact that the GeoLabel only utilizes the sequence information and lack of the use of the other information, we further developed the NetGO series tools based on GeoLabel to enhance the accuracy of protein function prediction. NetGO innovatively incorporated protein interaction network from string. NetGO collects label information from network to achieve protein function prediction. Experimental results have shown that the NetGO method is very helpful to improving the prediction of biological process and the cellular component ontology. Following that, we further introduced the LR text method and the sequence method on the basis of NetGO. LR text utilizes TF-IDF and a document to vector to encode protein-related scientific literature, uncovering information related to protein function in the literature. Sequence is a deep learning model which uses LSTM to encode protein sequences and utilize deep learning to explore the relationship between proteins and function. NetGO 2 participated in the CAFA 4 competition. According to the organizers, NetGO 2 from our lab once again achieved outstanding performance, obtaining the highest Fmax in MF, BP, and CC. It's worth mentioning that in CC ontology, our method outperformed the second place by 5%, which strongly confirms the effectiveness of literature information. Recently, it's impressive to see the evolution of protein language models, the replacement of the sequence method in NetGO 3, in NetGO 2 with the ES1D based method, which is protein language models and has proven to be highly effective in prediction protein functions, surpassing some previous methods. Integrating it into the NetGO 2 has resulted in the latest software method, NetGO 3. If you are interested, you can find more details in our labels. Next, we want to make a summary about this competition. In this competition, our methodology was developed on the basis of NetGO 3 and in integration. finally completed through a ranking model. In brackets, in the final prediction stage, our method only takes 2 hours to complete the process. It can be seen that our method is still relatively efficient in the final reference stage. Of course, in order to achieve better prediction, we have also designed several new component approach. Highlighted here are the latest design methods. Among these new methods, GeoXML and GeoRetrieval are based on textual information. LMNEM is based on both sequence and text information, and FORSEEK is based on protein structure information. Specifically, GeoXML leverages information from protein-related scientific literature to enable large-scale multi-label classification, different from the previous method. The SOTA method in stream large-scale multi-label classification is used here, attention XML, to find a protein-related functional information from the literature. GeoRetrieval is a new two-stage deep learning method. It takes into account both protein description text and scientific literature information, and looks for geo terms related to this literature information. LMEM combines the 3 types of information by collecting the relationship between protein literature and function in Geo and the GeoA database to jointly discover links between proteins and functions. FORSEEK is based on the similarity between the structures of proteins from FORSEEK and completes the functional prediction based on proteins with similar structure. When integrating these new component approaches, we also took into account the species information of the protein. At the time of integration, each protein is characterized not only by a predicted score from component method, but also a 21-dimensional vector to encode the species information. Specifically, there is a 21-dimensional one-hot encoding vector, where the first 20 positions represent the 20 most frequent species in training data, and the remaining one represents the other species. Eventually, the feature vector of each protein consists of the prediction score of the component method and this taxonomic vector. This figure shows the scores of our methods and the final model on public leaderboard. As you can see, the text-based methods on the right performed well in this competition, achieving significantly high score than the other component methods. We believe that that's why GoCurator is so successful. So now let's take a look at some of the best component methods in GeoCurator and see which one works well. In the courses of the competition, we found that protein 3D structure information and methods utilizing protein language models and methods utilizing biomedical taxonomy usually yields good results. For example, ESM1B, ESM2, and PROT-B5 all demonstrate good ability in predicting protein function. Using taxonomy methods, we mainly find out that the implicit connection between proteins and functions from the description text of protein-related scientific literature and the definition text belonging to geoterms. For protein 3D structure, we found that the protein with similar structures to test the proteins by comparing the similarity of structure. Finally, we get a prediction of the target protein through the function annotation from the similar proteins and get a good result in public leaderboard. For protein struct language modeling, corresponding dense representations are available given a protein sequences. With the help of functional annotation data, we can then predict the corresponding geoterms for these proteins. During the competition, we tried three methods, ESM1B and ESM2 and Prote5, and also consider concatenating their representations for prediction. Based on the performance of these methods, on public leaderboard, we found that ES1B is a better choice compared to the remaining two language models. We also noticed that the concatenating of the representation of three models results in better prediction performance. At the time of the integration, given complexity of the models, we still only considered predictions from ES1B, incorporating only this one protein language models. Next, we will describe each of the literature mining-based methods in detail. The first one is the GeoXML, similar to deep text Go method which applies literature to protein functions before. GeoXML tries to find the protein function-related information from its scientific literature. Specifically, based on the relevant literature information of the proteins in UniProt, we first encode these tests with BioBERT, and then utilize the attached XML to complete a large-scale multi-label classification. Finally, we unifying the functional information of proteins under literature by consensus methods to complete the functional prediction of target proteins. LMEM, on the other hand, considers the sequence description and the related literature information of the proteins at the same time. We noticed that some entries in the GEO and GEO database record the functional information of some proteins under specific literature, which help us to find out precise functional information from the literature. As shown in this figure, we encode the protein sequences, description, and publication information by specific tools. Specifically, we use Spectre to encode our literature, PubMedBird to encode description text, and ESM1B to encode amino acid sequences, and compile the encode representation to finalize the prediction of function. Geo retrieval, on the other hand, takes into account the textual information of the proteins and then considers the definition text of geoterms. The textual information of each protein is regarded as a query, and the text of geoterms is regarded as a document to find out the most relevant functional information on the proteins by information retrieval method. Specifically, we initialize search for possible candidate sets through the protein descriptions. Further, we utilize the document information of proteins and the definition geoterms to retrieve fine sorting, ranking the relevant functions at the top and eliminating the irrelevant ones, thus completing functional annotation of proteins. To summarize, each of these text mining methods utilize different text informations, and compared to the most basic text method, AI text, there is a significant improvement in efficiency. Regarding for training methods, we mainly introduce the following aspects. When training the model, we collect annotation data from three databases, Swiss Prot, GOA, and GEO. When dividing the data set, we consider two ways of division. The first one is the split based on time according to CAFA, And the second one is the sampling method based on species distribution. We sampled data from dataset based on the proportion of each species in the test set to obtain a validation set containing of 1,000 proteins. We found that the method of applying species distribution can get higher scores on public leaderboards. In the courses of the competition, except for taxonomy and taxonomic vector, we also noticed some interesting and important phenomena. How to integrate the net protein-related protein-protein interaction network into a final model is important. NetGO is a protein. NetGO looks for neighbor proteins of target proteins in the PPI network and predicts the function based on these neighbor proteins. However, we have found that the network information of some species may not be very reliable, and at the same time, some species may not have network information at all, which creates a very interesting phenomenon. When integrating NetGO into the model, we may not as good as not having NetGO at all, as scores that include NetGO are lower than scores that don't. Considering the property of PPI network, we believe that NetGO may only be suitable for predicting proteins from well-studied species. At the time of integration, proteins of most frequent 15 and 14 species were considered, respectively, with 15 species being considered for optimal performance. In fact, introducing species encoding into NetGO tree would give you a good score. That is, instead of the various neural methods we mentioned before, only a 21-dimensional taxonomic vector is introduced at the time of integration. We later submit NetGO tree predictions, and we end up with a score of 0.585 on the private leaderboard. Overall, NetGO is a powerful framework that can integrate multiple types of information and methods, and that's why GeoCurator works so well. Here we have listed some databases and tools that collect and process various information, which I believe that will be helpful for you. During the competition, each members of our lab played their roles, dedicating significant time and effort, and I'm very grateful for their contributions and the guidance from our mentor. Okay, that's all for today's report, and welcome for your questions.
Okay, so I'll read out the first couple questions that have come in the chat. We can also make it where people can feel free to unmute if they would like and ask their questions as well. So the first question that got asked was approximately what is the portion of proteins among the final CAFA test set that have literature information, i.e. text data? If you happen to know that. I don't know if they released that, but if you happen to know that.
Yes, I have seen the question. Okay, and you can see the answer from my supervisor. He has answered this question. So I repeated it again. The text set in the leaderboard is not released, but more than precisely and more than 19% proteins in the test supersets have literature information.
Okay, And the follow-up question was, could you explain how fold-seek KNN was implemented? Is it similar to BLAST KNN, where each template is weighted by the bit score?
Yes, yes. It's a similar method to basking. We use fold-seek to get the similarity score between text proteins and the proteins in training data. So we can make a functional chat. And we can transfer functional from training data from training proteins for target proteins.
Okay, while we're waiting for some other questions to come in, I guess maybe I can try to ask a question. Actually, I might need a second to gather my thoughts. I don't know if anybody else has any
Okay.
questions or if our other co-host Tianyu has questions. I just want to make sure I'm kind of thinking about this in the right framework before I propose it. But yeah, also feel free if anyone has any questions to go ahead and unmute if you don't want to put it in the chat.
Okay,
So I have a question on the net KNN. So you conclude that all using net KNN for all species is worse. I wonder whether this is because you only use Go annotation from Uniprot. So because only a small set of string entry can be mapped to Uniprot, and where only subset Go annotation. And if we use, but on the other hand, the string database by itself, it also has Go annotation directly to the string entries. So if you directly use the string database Go annotation, will you expect it to be performing better for a less studied species?
Okay, I think you have put out a good idea. Yes, we only use the annotation from Unicode, and we believe that the annotation from Unicode is more valid because the annotation we use is only experimental valid. We have not tried the annotation from the string databases. Maybe we should use it later to try to figure it out.
My question was going to kind of be along very similar terms to this previous question. Are there other databases that y'all were considering incorporating next year if you guys, assuming you guys attempt to do CAFA 6? Or do you think that the information that you get from the Go terms in the Uniprot database is sufficient for that portion of your method?
Yes, we're using the data. We're using the data following the previous CAFA setting, and we only use the annotation from these three database. Maybe we think it's sufficient for CAFA competition. Maybe in the future work, we should try some other databases.
So I want to ask like kind of philosophical question on the use of text data. So actually, I'm not really clear why text data will improve that much, because if the text, the literature is already used for annotating the proteins, then why would text mining be able to find additional Go terms?
We think that protein-related texture information is helpful for predicting functions. We realize that most of the proteins in the text set have annotated literature in this report, and specifically, some functional information about proteins can already recorded in their related literature. So we think it's crucial to utilize text mining method from this to figure out the most related functional information from the literature data. And we also
So I guess my question is this. So the experimental Go annotation which you use as the training testing, they come from literature, right? So, and then you also use the same literature to train your model. So I feel this is kind of circular logic.
We just use it for training, and we believe that in this way you can find the functional information from this experimental widely valid literature data. So when testing, if test proteins have some related literature data, we believe that the trained model can help us to find the relevant functional information for these test proteins.
So we had our previous speaker that we had, because we've been having a couple different protein function prediction kind of theme talks in the last few weeks. So our last speaker kind of presented a contrastive learning framework for predicting enzyme classes. So somewhat similar, but somewhat different from the way you guys are doing your learning and then doing your predictions. But do you think a contrastive framework could work for this type of data going off of Go annotations, or not as likely, and why or why not?
Oh, sorry, could you repeat it again? The
Sure.
connection to this is not stable.
Yeah. So our previous speaker was presenting also on protein function prediction, but they were doing it through the proxy of enzyme classes versus like Go annotations for a function. And so they used a contrastive learning framework to do that. So I was wondering, do you or your professor or co-authors who are in the audience think that a contrasting learning framework could work for this type of evaluation, and then why or why not? Yeah, it seems like your professor said maybe it just came out in literature too late, but I don't know. If you're familiar with the paper, and if you're not, it's okay, we can move on. But okay, it seems like he thinks that it could be something useful y'all incorporate in the future. So cool, good to know. Okay, I'll open it up just for like one more minute if we have any other questions. Any final lingering thoughts from anybody?
In the literature, some are experimental, some are computational. Do we have a way to differentiate computational paper in the text data?
We didn't make a weighting. We tried the same.
Okay. Well, I want to thank everybody let me just read. There are some top competitors using this technique. So, okay, cool. Good to know. Maybe that will be something we see from you guys next year. Well, again, I want to thank Shaojun as well as his professor for being here with us today. We will have the recording available on YouTube. In a couple weeks, we also have someone who is partnering with us to put this on Bilibili if you're in our audience and interested in seeing it that way versus YouTube. So yeah, that concludes today, and our next talk will be in 2 weeks at our regular time. So thank you so much, Shaojun, and thank you so much to everyone who attended.